Hi, this is part of the series www.resetonline.org. This is the fish and plants experiment. This is the second session in that experiment. It's not a standalone video, but this is the procedure that we'll do at the end of this experiment 6 to 8 weeks after it commenced. If there's time in the first session, it's helpful to introduce the concept of wastes and nutrients so that students can get a better sense for what might be happening here in that wastes have nutrients in them, and those nutrients can help plants to grow. Can introduce the concept of fertilizers, such as fertilizer that comes from cow manure or horse manure, pigs, chickens. And farmers use that on the fields to help the plants to grow. This often will introduce the yuck factor to students, but science is often yucky, as I tell them. So at the end of this experiment, we will have had, over the course of the 6 to 8 weeks, one plant from each of the groups of 4 to 5 students in this container, which just has tap water in it. It'll get the same lighting, it'll have the availability of water, but it does not have the access to nutrients that these plants will have. In here we have our betta fish that's been fed over the 6 to 8 week period. The plants will have had the same access to light and water, but they'll also have had access to nutrients as well during that time frame. I had one of my students answer this questionnaire that I give at the end of all of my science sessions with that particular group. And this question is, what experiment did you like the best and what did you learn from it? And his answer is the fish. I learned that fish poop. Well, I hope that's not all that that 4th grader learned, but that was part of this experiment. So the procedures here is I'll have each group of students get one of these plants, dry it, and weigh it. They'll use the scales that they learned how to use at the first session, and they'll calibrate it so it's 0, starts at 0, and each group will weigh one of the plants from this container. It need not be the same group same plant that group weighed in the first session, because we're dealing in terms of comparison of change in biomass with the sum of all the weights of the plants from this particular vessel. So I'll have the students record that weight on the same data sheet that they used the first time around. It's important to safeguard those data sheets because they tend to disappear if you leave them with the students. And then I'll add those, and we will take that figure in grams, and we'll subtract from that the weight of all the plants in this vessel at the beginning, the sum of the weight of all the plants at the beginning of the experiment. And that will represent the change in the biomass weight over the course of the experiment. Bring those plants back up front. Send the plants from this vessel that has the Siamese fighting fish, or the betta fish, in it, and has had the access to those nutrients over the course of the experiment. Have each group of students will weigh one of these plants, check the calibration on the scales, weigh, record on the data sheet, and we'll do the same thing in terms of adding up the weight of the plants, and then looking at the sum and subtracting from that the sum of the weight of the biomass at the beginning of the experiment. And if things went according to plan, these plants will have gained more biomass during the experiment than did these, which did not have access to nutrients. I then used this experiment to introduce the concept of water pollution to the students. The vocabulary I used in this final session, after reviewing the vocabulary from the first session, which included nutrients, energy, photosynthesis, scales, and calibrate. The words that we'll use in this final session of this experiment: pollution, algae, eutrophication, and dead zone. So I explained to the students that some wastes in water can be helpful. We found out via this experiment that the waste from the fish help the plants grow. So not all wastes in water are pollution, but too much waste in water causes pollution. Talk about the sources of pollution in water bodies, and it can be nutrients that come from human waste, travels to the river through the sewage treatment plant, but some of the nutrients end up in the water body. Can talk about nutrients from farms, where farmers apply fertilizer to the field, and some of it will wash off with the rain. Also from feedlots, where cows and pigs are kept. Their manure, too much, can wash off into the water more than the system, the water body can handle. And that's where I introduced the concept of algae and algae growth, algae being tiny plants. And when there's a lot of nutrients coming into the water, these plants grow and grow, and too many of them grow, then they die. And as these one-celled organisms die, they use up the oxygen in the water, and that causes the problem of oxygen deprivation. And I don't use that word with these students, but the lack of oxygen in the water body, that then causes organisms to die, or a minimum fish have to leave those waters because there's not sufficient oxygen for them to survive. Those are the lessons that I use. I also use the term dead zone. In many of the bays around the United States have dead zones in them. They're called dead zones because they lack growing organisms because of the deficiency of oxygen. Sometimes this experiment does not come out the way that it's planned. I've done it myself at home, and it has worked, but sometimes the results I get in the classroom are different. And I think that's a learning experiment. I can ask the students what might have gone wrong in terms of it not coming out the way that we had supposed it would come out, and what might we be done differently. Of course, one hypothesis is that the fish was nibbling on the plants, and that caused the biomass to decline or not to grow as much. I happen to know that this particular fish doesn't feed on these plants, but it's a great hypothesis to discuss. But I also discuss things that might have led to results that might not be correct, such as these scales, which are very rough, may not have the refinement of reading, the accuracy of the reading to detect the changes, slight changes in weight that might occur in these plants across the span of this experiment. It could be that the way we read the weights was not correct. And then I'll introduce the concept of what else might we try with this experiment to make it work better next time. And one would be to use more refined weight or scales that read more accurately. Another might be to run the experiment for a longer amount of time, such that the change in biomass would be more significant than we are able to find in the 6 to 8 week period. So that's the concluding video for what I think of as nutrient cycling, but don't use those terms with most of these students, but the plants and fish experiment.